I have a great pleasure to introduce the next speaker, Professor Annette Ziegler from the Helmholtz Center in Munich, in Germany. And she's the director of the Institute of Diabetes Research at the Helmholtz Center. She has also other major titles. She's the chair of diabetes at the Faculty of Medicine at the Technical University of Munich. She's a speaker of the Global Platform for the Prevention of Autoimmune Diabetes, as well as an academy head of the German Center for Diabetes Research. And Annette has made several truly seminal findings in understanding the pathogenesis of type 1 diabetes and bringing the findings to the clinic. These include, for example, her truly groundbreaking discoveries on the start of islet autoimmunity already during the first year of life, and insulin as the initial target of autoantibodies. She also founded a global platform for the prevention of type 1 diabetes, in which she introduced polygenic risk score-based newborn screening at the population-wide level. And this has then paved the way for prevention trials, where her goal is nothing short of finding a preventive treatment for a world without type 1 diabetes. So thanks so much for doing this talk, and I really look forward to hearing.
Yeah, thank you so much for inviting me to this fantastic meeting. Congratulations, actually. And I'm a little bit exotic because you heard already that I'm speaking about type 1 diabetes, about childhood diabetes, and here are my disclosures. And I think you may know that type 1 diabetes is a metabolic disease, but it is also an autoimmune disease. And autoimmunity is actually preceding the clinical manifestation of type 1 diabetes. And during my talk, I will speak about the initiation of this autoimmunity, why and when does it happen, but also how we can diagnose autoimmunity on a broad populational level, and then briefly also about our ways to try to prevent autoimmunity in clinical trials. So I think the first important thing is that we have a biomarker to detect autoimmunity, and these are the well-known islet autoantibodies. And we know, and I think that was really very important work following over 20,000 children from birth, we learned that if a child develops more than one of these islet autoantibodies, namely 2 or 3, then they will develop type 1 diabetes, clinical type 1 diabetes, during lifetime. And therefore, this has really led to some kind of paradigm shift in guidelines relative to type 1 diabetes, namely stages have been introduced. And the stage 1 and stage 2 reflect this early stage of autoimmunity where there is no metabolic symptoms. The child or the person does not know that it has this clinical disease. It is just the autoimmune, the antibodies that are present. But we call it the start of type 1 diabetes, stage 1, just with antibodies, stage 2 with form of impaired glucose tolerance. And what one in the clinic sees with symptoms is only then stage 3 type 1 diabetes. And also what is very, very important for the autoimmunity being really relevant is disease is that we now have a first therapy that can delay this disease when given at stage 2 type 1 diabetes, namely a single course of 2-week antibody treatment with teplizumab, an anti-CD3 antibody. CD3 is expressed on T cells. And you can see here that if you give this for 14 days, then after 18 months is the biggest effect, only 5% progression to insulin-dependent diabetes compared to 45% in the placebo group. And you can also express this by delays by years, and we know that it delays the clinical onset of disease by 2 to 3 years. For us in type 1 diabetes, it is a milestone, it is a breakthrough because it is the first therapy that is now approved in America, not yet in Europe, as sealed. And this therapy is addressing the immune part of that disease. And so far, we have only insulin treatment for type 1 diabetes. So it's the first immune therapy and also the first therapy to delay this disease given to people at this early stage. But of course, and I come to this a little bit later, that also calls for the necessity to introduce broad population-based screening for the autoimmunity of that disease, because this drug is given at an asymptomatic stage. So nobody knows that it has this stage without participating in screening, and that is why this becomes actually very important. But we have asked, and I will first talk a little bit of why this autoimmunity happened and when it happens. And that was, I think, one kind of important, relevant finding when we followed these children and babies in these cohorts from birth, that we found that this autoimmunity is not happening at the same rate during lifetime, but there is a peak incidence very, very early in life, around 1 to 2 years. And on the left side, you see this in our first study that I was organizing in Germany. Baby Diab is a study of offspring of people, parents with type 1 diabetes, either father or mother. But it was also seen in the large international TEDDY study that follows people from the general population. And so just both studies confirming that there is this early peak of here islet autoantibody seroconversion. And of course, that is then let us ask the question somehow, why is early in life such a susceptible period for this autoimmunity to start? And the questions that we had, of course, do we know what triggers the antibody, and are there other markers or other something else that precedes the development of autoimmunity? So, of course, very important is the genetics. And here you see many genes, and there are many more now that have been described to be associated with the development of autoimmune diseases, in particular type 1 diabetes. And the blue one is the largest one, HLA, but there are many others, and most of the genes are actually relevant for immune response. But there are also some metabolism genes like insulin that is associated with an increased risk to develop type 1 diabetes. And what we have done, and it was mentioned before, that we have combined all these genes in a polygenic risk score. And what we can do is actually that we can stratify risk by using this risk score with all the genes that have been described together with HLA in the general population, and identify people that have a risk of more than 10% to develop this autoimmunity within the first 6 years of life. And comparing to the background population, you all know type 1 diabetes is not as frequent as type 2. It is about 0.4% of people developing type 1 diabetes. So it's 25-fold increase just by using genetics. But also, what we have found is that there are inflammatory signatures that precede the development of this autoimmunity. And one of these signatures is a type 1 interferon transcriptional signature that we described of lymphocytes of these children. And I think you can see here that before they develop on this AAB, means these antibodies, they have a slight increase of this signature, but interestingly, this signature also correlates with the number of infections these children had. Namely, if they had 2 infections, the signature before that time, the signature was higher. So inflammation is somehow relevant, and you see this also in a second approach where we looked at T cell responses to insulin. You have heard before that insulin is one of the primary target of this autoimmunity in the children. You see here the gene expression of this insulin reactive cells in a control group who stayed healthy, and compared in people who developed these antibodies already at 6 months of age. So also very early there is already a kind of propensity to a Th1 immune response in these children. So genetics and inflammation is important in the autoimmune disease, but I know that you are interested in metabolism, and I just want to show you that there is also metabolism and islet function who are probably very relevant for this disease. I have forgotten one slide. So if we link actually this type 1 interferon signature, that you can also say it's a cyclic expression on monocytes, together to the CD4 T cell response, it really looks like that CD4 T cells react more under an inflamed condition with a cyclic monocyte positivity. So probably this inflammation and the T cell response somehow go together. And coming back also to the virus again, there is in TEDdy study shown that virus and positivity for even 2 viruses are actually associated with an increased risk. Viruses found and sequenced in stool samples prior to autoimmunity to develop this autoimmunity. So that all looks like maybe infections, virus infections, inflammations kind of trigger the autoimmunity. But now glucose metabolism. What we also did, and we were actually very lucky by chance because we did one of these intervention studies using oral insulin to sensitize against the insulin, to try to somehow train the immune system to accept the insulin. And by doing this and giving children this oral insulin, we check the glucose levels constantly because, of course, you have also some fear, although if you eat insulin, it's not metabolically active, but you want to make sure that the children and babies don't develop hypoglycemia. So we checked glucose values, and then we looked at these glucose values in people that were enrolled in this study. They were the majority healthy individuals. And it was kind of surprising, and by the way, it is also striking that normal glucose values are not published yet, and this is just a publication from last year, that we saw that glucose in the first years of life changed quite dramatically with a decrease very early in life in the first year and a increase later on. And somehow this glucose levels, they coincides with the peak of autoimmunity or mirror this peak of autoimmunity. If you remember that there was a peak very early, and it's kind of almost looks that the glucose go the other way. So islet activity seems to be very high in this susceptible period. And I would also, so I jump a little bit from theme to theme, but I just want to tell you that metabolism is important in type 1 diabetes, that there has been 2 elegant Mendelian randomization studies looking at genes that predispose for childhood obesity, and those also predispose for the development of type 1 diabetes, also underlying the fact that obesity and growth and childhood growth are not kind of neutral, and they also may play a role. And if you look again at this glucose, then if somebody of you may know that the growth curve of children around that time is actually also just kind of the opposite or preceding a little bit this glucose nadir. So another finding is that a child, and this is totally opposite to type 2 diabetes, a child that is born to a mother with type 1 diabetes is somehow protected from this immune first wave of autoimmunity, what you can see on the right side here. So if you have a mother with diabetes compared to a father, you have a lower peak incidence of this autoimmunity, and the peak is also somehow later. And on the other hand, as an offspring of a mother, you have higher insulin levels during the first year compared to non-diabetic mother. So again, metabolism changes somehow autoimmunity risk. And the last point I want to make, that we also looked at these glucose levels relative to the appearance of autoantibodies. And it was somehow surprising and unexpected to us that those children who were antibody or developed antibodies later in life, here in blue, they had already slightly elevated glucose levels. By the way, we have excluded every children who had developed type 1 diabetes during this early period. So the glucose levels in general were within a normal range, but in those with antibodies were already 4 milligram per deciliter higher. And if we looked at the 30 minutes postprandial levels, they were 6 milligram per deciliter higher. So we thought, okay, let's look at this relative to when the antibodies appear. So we kind of aligned everybody to seroconversion when they developed autoimmunity and looked at glucose levels. And what we found is that the postprandial glucose levels somehow started already to rise before the autoimmunity came, while the other line, the preprandial, started to rise a little bit later. And here the difference to the negative line, and the negative line are those who stay healthy and don't develop autoimmunity, are 15 milligram per deciliter difference. So that actually suggests that metabolism change in glucose precedes the start of the autoimmunity, and that could explain that there is somehow of an islet insult happening, maybe a virus, and that makes the glucose rise and the autoimmunity to start. So but just telling, while I was saying before, type 1 diabetes is an autoimmune disease, is preceded by autoimmunity, now we have to think maybe also preceded some defect in metabolism. Okay, so the message of this part is inflammatory signatures in immune cells primed toward an inflammatory response are found in children before they develop autoimmunity. And there are, and mainly prolonged and multiple early viral infection increase also the risk of autoimmunity. So there is a link of this inflammation to maybe virus infections. And metabolic alterations like the rise in glucose are present earlier in the disease process than we have previously considered. And the sudden rise in postprandial blood glucose shortly before seroconversion may be consistent with an event that affects islet function, and that could be a virus. So one other, coming back to the metabolism, is that now when we're looking at cross-sectional population screening, and this is where I'm coming now, and who develops this autoimmunity in these children, these are not over 90,000 children in Bavaria, Germany studies, also obesity comes out as a risk factor for autoimmunity. And that is not only in this study, it has been shown in many other cohort studies. So just also underlying that maybe the link to obesity and autoimmune diseases is also kind of there. So this is just the combination, what I said to you again, what contributes to start of islet autoimmunity, genetics, infections, inflammations, and islet activity, or also vulnerability in that case. Now, coming to the second part of my talk, public health screening for islet autoimmunity. So I said to you, we have a new drug that can delay this disease. And we have to ask us, okay, how can we bring this drug to the majority of people who develop type 1 diabetes only by doing such a public health screening for early stage of the disease. And we have actually started that as a pilot project in Bavaria, Germany in 2015. And at that time, the treatment was not yet available, but the idea behind it was to improve care through early diagnosis, education, and treatment, and identify children who may benefit from immune-based therapies at that stage in clinical trials to prevent insulin dependence on a broad population level. So what was our approach? We actually did a model, and I don't have that here because of time reasons, but to see what is the best age in terms of sensitivity to do this screening. And that best age for a one-off screening was 4 years. So you pick up the most children who develop this disease, and you have already developed the autoantibodies that you can test them in a cross-sectional screening. If you do 2 screens, the best age is twice within 10 years. So when we started this study, we first did a 1 screen in children 2 to 6 years, and then we later developed it over the last 3 years into 2 possible screens. And we used a very easy screen in capillary blood, a commercial ELISA from RSR, a company in UK. And then if that gave a positive signal in the same capillary blood samples, we tested our 4 islet antibodies that are mostly used by our in-house assay. And I can tell you that I know that there's a similar project now in Denmark and Sweden called DiaUnion, I think. They use slightly different assays, but I think they also use home testing. But the idea behind it is very similar. And then if in both settings 2 antibodies were detected, we asked for a second venous blood sample, we confirmed the result, and then we said the child has a diagnosis of early stage diabetes and invited the children also for further staging, for education, for a training program, etc. And we have now expanded from Bavaria, Germany to Lower Saxony and Saxony. And in cooperation with pediatricians, we did not home testing. This was the pediatrician in the office who took this capillary blood, and there's over 600 pediatricians participating. We screened over 170,000 children in that age range, and we found 0.3% to have that early stage type 1 diabetes with at least 2 islet antibodies. So, of course, I didn't bring all these slides. The question is that beneficial? So the data, they are published, and we found actually in many aspects that it's good to screen. It reduced the rate of diabetic ketoacidosis dramatically from an average of 25% in Germany to less than 2%. And in the pandemic, where we also did that, it was even 40% DKA rate and was still 2% in that study. But we have also much better beta cell function at clinical diagnosis. There is actually a reduced stress and burden, and these people can participate, and they did participate in trials, and also actually in this teplizumab trial that is now available as treatment in America. So you may ask, how many have what stage? The majority has still stage 1 diabetes without any impaired glucose tolerance, but there are 10% who have that stage 2, this glycemia, and you detect also around 4 or 5% who have actually already overt diabetes, but in a still asymptomatic stage. So it is, of course, if you consult these families, important to know how fast, how quickly they progress to insulin dependence. And that was very important to see for us. So it is in stage 2, over 50% who progress to insulin dependence within 2 years. But in stage 1, it is much, much lower. But the data are kind of identical to previous studies in people with an increased genetic risk like TEDdy, or also in TrialNet studies from people with a family history of diabetes. So we can actually really say if you have a general population child with antibodies, your risk is identical to someone who has a family history with diabetes in terms of disease progression. So what we were also interested in, if you look at this relative low risk of those with stage 2, to see, because these were the majority of people, over 80% had stage 2, can we further stratify in this group progression to disease? And so we looked at various markers that actually are associated with disease progression. And interesting, that is again, disease progression is less autoimmune, but very driven by metabolism. You see, it is already within the normal range, kind of, but the HbA1c that they have, the 90-minute glucose level, also 120 works and is significant, but 90 gave the best performance. And it is also a little bit the autoimmunity, namely the titer of IL-2 antibody that matters. And with this, we created again a kind of progression score, progression likelihood score, by adding the risks of those markers. And by looking at this score, we found that people that had a score higher than the 90th centile, that these within the stage 1 had also a progression that was similar to stage 2, namely over 50% within 2 years. And I think that is very important, that we learn to further stratify the risk, because we want to advise the families clearly. And also, we want to make the treatments available to those who are at highest risk. And we would suggest that also this group, which we call now stage 1B diabetes, is eligible for the teplizumab therapy. So last part, very short, I would just like to show you what we are also trying to do, namely primary prevention. So what we want to do, that we would like to, by giving preventions very close to birth to children with the highest genetic predisposition, to prevent stage 1, 2, the whole autoimmunity to start. And to be able to do this, the genetic risk score was for us very important, because we need to stratify risk in the general population. And we founded this platform, European platform called GPAD. And this platform identifies newborns from different countries attached to the regular newborn screening, but as a research project, doing this genetic risk scores with a risk to develop type 1 diabetes above 10%. And those children and then asked and families to participate in trials trying to prevent the autoimmunity. That's the platform. These are the different countries and regions in Europe that participate at the moment at this, and we hope to be able also to extend this further with future trials coming. So this is this application of a 47 SNP risk score. We have screened over 400,000 newborns, and about 1.3% have that increased score. And these are the children where we offer the intervention. So our first intervention was POINT, the primary oral insulin trial. That was also the study what I mentioned before, where we measured the glucose. The idea is to give oral insulin to sensitize with insulin powder to train the immune system towards tolerance. Little bit like, you know, from allergy, peanut allergy, to give peanuts to children to avoid allergy. And so we had different doses. It's a 1-to-1 randomized trial. We needed 1,050 participants, babies. We give that from 4 to 7 months of age. That is when they enter in the study. They eat the insulin or placebo daily until 3 years of age. the enrollment is complete, but the trial is still blinded. It will be every children will have completed the treatment, also follow up until 6 years of age in 24, and this is when we unblind the study. You can also see that enrollment was actually very, very efficient. We enrolled 1,000 children faster than expected. And so at the end of 24, we hope to have the trial. The second trial is Cynthia, is a trial using a probiotic, and the idea behind that is that probiotics and the microbiome is also something that is changed in people who develop autoimmunity. And here we have several outcomes: diabetes, celiac disease, and also allergy, because it's a broader application. And also here we are still enrolling, but we have over 700 children now enrolled in the study. But I don't want to go into details, just say that this platform now exists. We plan actually the next trial for 24, and we hopefully find some treatments that can modify this course just from start. I would like to acknowledge all my teams from Helmholtz Munich, the FRIDA study team and all the pediatricians, the POINT and GPAD teams, and thank you for your attention.
Thanks for a wonderful talk. And now it's time for questions from the audience.
Thank you for, Marta was a group leader here at CVMR. Thank you for this enlightening talk and for your treatment of newborns with diabetes. My question is related to when you were pointing about the mother and the father and the relation that this would have with the children. And I was wondering if you have done some work also identifying if there are any risk factors during the pregnancy of the mother that could affect the treatment or the onset of the disease, or even like delaying this. This is more about thinking about the prevention, if there are any factors. I'm thinking GDM, obesity of the mother, or diet, or other factors that could contribute to the differences, especially on the stages that you were mentioning.
Thank you very much. We have not actually looked at that in detail, or people have looked at it, but it's not really consistent that something came out. But exactly, very important to look and to understand what the maternal protection is about. Because if we understand this, that would be a very nice way to treat maybe. And we are doing some studies in this, but I think we cannot really have a conclusive answer at the moment.
Thanks again. That was really excellent. My question is related to something you didn't talk about, and that's COVID. And so what do you expect the impact of COVID will be on the development of new cases of people with type 1 diabetes? Is there data today, or is it too early to say?
Yes, there is data, and it probably has some impact. So first of all, the data says that there is an increase in type 1 diabetes during the COVID time. But there is also now some new data that is not published but in archives, saying that it is really related to having the COVID infection that increases the risk of autoimmunity. We are also looking in this in very detail. I can't really speak about these results in detail yet, but I think it is also an enormous opportunity, because we have a COVID period where there is a virus that enters the beta cell. The beta cell has the COVID antibody receptor, and there's not many other infections in this time, because other infections really went down enormously. And my guess is we will see further increases in this disease through the pandemic. But in detail, it has to be proven, but I think there will also be more people looking into it and publications coming out.
Hello. First of all, thanks for a very interesting talk. My question is that you have been looking into type 1 interferons and in relation to viral infections, but have you also been looking into type 3 interferons?
No, I mean, this was a study of actually a transcriptome of PBMCs, and we only found this type 1 interferon signature be associated with the early onset. But I don't recall exactly whether type 3 interferon signatures were really looked at. I don't know, but it didn't come out.
Thank you for a fantastic talk. I have a question about type 1 diabetes in different ancestries. This comes from naivety on my part, not really having a good sense of the literature. Could you comment first off on to what extent you think you capture diverse ancestries in the studies that are ongoing in Europe? And secondly, to follow up on Julien's comment around COVID, do you think that COVID will have a different impact in different ancestries in terms of type 1 diabetes risk?
So what do you mean exactly with different ancestries in terms of?
So I mean that when we look at the frequency of type 1 diabetes around the world, it tends to be increasing in Western countries, but we don't see the same increase in prevalence in Africa or in other populations. I think that's important, but we have also people who have moved. We have immigration, we have people who move to in a different environment. So I'm just wondering what we can learn about what is the underlying reasons for why type 1 diabetes is more frequent in certain ancestries. Does that make sense?
Yeah, no. I mean, I'm not 100% sure whether we know enough about countries like in Africa or other countries, because, it is relatively recent that insulin was discovered and these people survive, if you want, and they survived certainly less well in poor countries for some time. And so these are 50 years, 60 years that we are now following this. And I think that there will probably also be an increase in other countries that we just don't monitor well enough yet, and it's hard to conclude. In terms of genetics, there is some change. It looks like that there are more susceptible genes now than it was very much HLA dominated. Now more and more other genes seem to play a role, and that could be also, of course, in response to environment and exposures. But I think it is not entirely clear yet.
We have time for one final brief question.
Regarding the interesting observation of children to mothers with type 1 diabetes has a lower risk of developing type 1 diabetes afterwards, can you speculate on the mechanism behind?
Yeah, so the spec is really a speculation, but it's twofold. So the one is more on the immune system, the other is more on the metabolism. So I showed you that the children have higher levels of circulating insulin in the first year of life. That could, of course, also theoretically improve tolerance to insulin if you have higher levels of a hormone very, very early on where the immune system matures. So improving tolerance to this antigen. The other is that we actually have done some studies in cord blood T cells, and we see actually some reactivity already to insulin in children of fathers with diabetes in the cord blood, and the children with mothers do not have that. They are looking very similar to absolutely healthy controls. So it may also be during the maturation of the immune system very, very early on that being exposed to the autoimmunity and that helps somehow to be trained early enough. But as I said, we don't have enough understanding yet to be really conclusive here. And to Anne's question also during COVID, I mean, I don't know whether different population react differently, and it will be probably also hard because, COVID was so different around countries, like with lockdown and lockdown exposures, to be really, to know whether it is the same mechanism everywhere. Yeah.
All right. Thanks for a great talk.